Assalamu alaikum. This is the continuation of previous lectures. We have discussed about the enzyme action. Then in second lecture we discussed about the effect of temperature on enzyme action, and in today's lecture we have to discuss about the effect of pH on enzyme action. Enzymes, organic catalysts made by living cells. Without enzymes, there would be need of very high temperature for metabolism. Enzyme and its substrate to form a short-lived intermediate enzyme substrate complex, and once reaction occurred, complex break into products and enzyme. Enzymes speed up biochemical reactions. For example, one molecule of enzyme catalase decompose about 6 lakhs molecule per second of hydrogen peroxide to form hydrogen and oxygen at body temperature. And it's very interesting that enzymes itself, they are produced in very small quantities but brings about greater changes. Okay, these are the outlines. Effect of pH on enzyme action, definition of pH, pH scale, optimum pH of different enzymes, effect of pH on different enzymes, graphic or illustrations, images. Enzyme work on specific pH. Its activity varies considerably with pH, and there is generally a marked optimum pH for each enzyme. Now question arises, what is pH? Biology is very interesting subject. In biology there are different concepts. If you have a clear concepts, then you can understand any topic and interest will grow up. So I think I should have to define what is pH. And the definition of pH is the negative logarithm of hydrogen ion activity in a given solution. It's a potential hydrogen. pH stands for potential hydrogen, and pH scale measures how acidic or basic substances. In logarithm, the pH scale ranges from 0 to 14. A pH of 7 is considered as neutral. A pH less than 7 is acidic, and greater than 7 is basic. This pH scale shows acidic condition, alkaline condition, and neutral conditions. It's an illustration that shows pH scale. It shows that there is range 0 to 14 and 7. At 7 you can clearly look at this pH scale. At 7 whenever there is 7 pH, it means the solution or substance is neutral, and more than 7 there will be weak strong, then gradually it will weak, and finally it become a complete basic. And as far as whenever going down, weak and acidic medium, and at number 1 it represents very strong acid. It means more hydrogen bonds means very strong acid. Fewer hydrogen bond means any substance, it is alkaline in nature, right? It's a pH scale. Every enzyme function in a particular range of pH when temperature remains constant. The maximum rate of reaction occurs at optimum pH. When pH changes to above or below this value, enzyme change its function. Optimum pH means it's a narrow range of pH in which an enzyme can work most effectively, right? For example, optimum pH of some important enzyme, as for example pepsin, its pH is 2, that is very strong acid. Amylase, its pH is 6.80, that means it's near to neutral condition. Sucrase, it's 4.50. there is graphic representation. It shows that different enzymes have different pH. Pepsin, there is illustration shows the pH of pepsin, salivary amylase, and trypsin. So pH is a measure of the concentration. There is another chart that shows the different pH of different enzymes. For example, pepsin, sucrase, enterokinase, salivary amylase, catalase, chymotrypsin, pancreatic juice, and arginase. Actually, pH is a measure of concentration of hydrogen ions in a solution. Lower the pH, higher the hydrogen ion concentration. Hydrogen ions can interact with the R group, that is radical group of amino acids, affecting the way in which they bond with each other and therefore affect their 3D arrangement. A pH which is very different from optimum pH can cause denaturation of an enzyme. Now, what are the effects of pH on enzyme action? Effect of pH on enzyme action, number one is a slight change in pH can change the ionization of the amino acids at the active site. As a result, enzyme activity is either retarded or blocked completely. And secondly, extreme change in pH can cause the bonds in the enzyme to break, resulting in the enzyme denaturation or the destruction of enzymes. It's a graphic representation. Rate of reaction shows effect of pH on the rate of enzyme control reactions. Most enzymes have optimum pH. This optimum pH is between 6 and 8. When pH is too low, the hydrogen ion combine with the radical group of enzymes amino acids, so it reduces their ability to bind with substrate. Acidic environment can also denature enzymes. Attachment of cofactor changes the shapes of the protein and allows it to combine with its substrate. Right? Every enzyme functions in a particular range of pH when temperature remains constant. The maximum rate of reaction occurs at optimum pH. When pH is changed to above or below this value, the enzyme changes its function. As shown in this table, there are different enzymes which shows different optimum pH. Pepsin work when the pH will be 2, means पेप्सिन को काम करने के लिए अगर pH 2 है, इसका pH 2 है तो पेप्सिन उसमें वर्क करेगा. अगर इससे कम हो या इससे ज़्यादा हो तो पेप्सिन का वर्क नहीं होगा. सक्रेज को रिक्वायर्ड है 4.50, एंट्रोकाइनेज को रिक्वायर्ड है 5.50, स्लाइवरी एमाइलेज को रिक्वायर्ड है 6.80, कैटलेज को रिक्वायर्ड है 7.60, काइमोट्रिप्सिन को रिक्वायर्ड है 7 to 8, पेनक्रेटिक जूस को रिक्वायर्ड है 9.00 एंड आर्जिनेज को रिक्वायर्ड है 9.70. अब ये चार्ट, इस जो टेबल है, ये इस चीज़ को शो कर रहा है कि एंजाइम, हर डिफरेंट एंजाइम का डिफरेंट पीएच है और वो अपने पीएच पर ही वर्क करेंगे. राइट? तो अब इसमें अगर आप देखें तो पेप्सिन जो है वो एसिडिक मीडियम, स्ट्रॉन्ग एसिड, और अगर आप इसमें देखें पेनक्रेटिक जूस को देखें या आप आर्जिनेस को देखें तो इनके ये बिल्कुल एल्कलाइन मीडियम में. तो ये किस तरह से हो रहा है कि एसिडिक मीडियम में इनके वर्किंग किस तरह से है? तो इसका आंसर ये है कि पेप्सिन हैज़ एन optimal of 2.00, that is its pH, required pH. Pepsin has an amino acid sequence. This sequence maintains its ionic and hydrogen bonds, so it can function at such low temperature. Right? On the other hand, trypsin, it is active in more basic medium, that is pH 9. This pH is found in small intestine of mammals. The optimum pH of enzyme shows the pH of the body fluid. Enzymes are found in this fluid. So we can say that different enzymes have different optimum pH, and it is the end of today's topic. Inshallah, we will continue this chapter in the next lecture.